right. Thank you very much for being here. I think Paula is very popular. It's going to be a very popular talk with many people calling remotely. It is a great pleasure to introduce Professor Tor Wager. Tor Wager from Dartmouth College. So this is the first of the beginning of this year's BIRC Speaker Series. We experimented last year with several sites on doing a remote WebEx call, and it was quite popular with about 10 to 20 people per sites calling in. So we expanded it more to over 40 sites. We'll see how it picks up. But hopefully a lot of people will log in and get a chance to hear people talking, such as Tor, which is a huge treat for us. So Tor is the Diana Taylor Distinguished Professor in Neuroscience at Dartmouth College. He received his PhD from the University of Michigan in cognitive psychology in 2003 and served on faculty at Columbia University and at University of Colorado Boulder. Since 2004, he has directed the Cognitive and Affective Neuroscience Laboratory, a research lab devoted to work on the neurophysiology of the affective processes, pain, emotion, stress, and empathy, and how they are shaped by cognitive and social influences. Dr. Wager and his lab are also dedicated to developing analysis methods for functional neuroimaging and sharing ideas, tools, and scientific data with the scientific community and public. I myself have spent in our lab and benefited from his tools and scripts as well, such as the mediation toolbox, among others. And so we're very fortunate to have his presence in the scientific community. He has over 200 publications that are widely cited, including journals. I'm tempted to read all of this that's written here, but it's going to be quite long, but it includes Science, Nature, Neuroscience, Neurons, Nature, Methods, PNAS, Psych Science, Past Biology, Trends in Cognitive Sciences, New England Journal of Medicine, Nature Reviews, Neuroscience, Nature Medicine, and Journal of Neuroscience. He's currently a PI or multi-PI in five major NIH grants, and he has received a number of awards, including the McLean Award from the American Psychonomic Society, the Rising Star Award from the American Psychological Society, and the Cognitive Neuroscience Society Young Investigator Award. And you have two patents. You're going to be talking about major patents, or is that
a little bit?
Just a little bit. And he is the past president of the Social and Affective Neuroscience Society, Council of the American Psychonomic Society, and the secretary of the organization for brain mapping. So we have a very accomplished scientist here. We're very fortunate to have you here. I follow his tweet, and one thing that I've noticed is his tweets are very thoughtful and warm, and I could tell that you were a nice person even before reading it. You should follow his tweets, and you'll get some great thoughtful insight from his tweets. So it's a great pleasure to have you.
Thank you. Thank you all for coming. It's really an honor to be here to be able to discuss our research with you. I'm going to work on my tweets, but I'm a nice person. There we go. Now it works. Awesome. Thank you. This is the Ebers Papyrus, circa 1500 BCE, which is one of the oldest known pharmacopoeias in modern times. And in it, you'll find descriptions of over 400 ancient remedies, some of which we might recognize today. If you have a headache, you might be prescribed wormwood, which is related to absinthe, and opium. If you were poisoned, you might get something different. Powdered emeralds, or other precious stones, or bezoar stones, which is undigested matter from the intestines of animals. And these treatments continued, many of them throughout centuries, well into the 1900s, and some of them even today. They included things like human sweat and feces, tapeworms, and other kinds of worms. One of my favorites is Actia, moss scraped from the skull of a hung criminal. And later in 19th century Europe, powdered Egyptian mummies. These treatments were given to the wealthiest and most privileged among us, as well as others. You consider the case of King Charles II, who had an undiagnosed ailment treated with bleeding first, which is very common. So a pint of blood extracted from his right arm and a half pint from his left shoulder, then an emetic, physics, and an enema with various substances. The royal head was then shaved and a blister raised, which you can see an analog of in modern-day cupping in the Olympic other sports competitions. Sneezing powder, emetics, bleeding, pitch and pigeon dung on his feet, potions with different substances, 40 drops of extract of human skull, and the application of the bezoar stone, after which His Majesty died. And the surprising thing to me about this is not that there were so many ancient medicines that were tried, right? But that their use persisted for so long. How could we not figure out which of these things were actually harmful or deadly to patients? We think, of course, that times are really different now, and we're in much better shape, but that's not necessarily so. This has been a trend throughout the history of medicine. We have more procedures and less care, which is in part what prompted Frank Peabody many years later to write, "The secret of caring for the patient is caring for the patient." Maybe there's something about these ancient treatments and how in the care and the ritual provided by the physicians and the healers that was actually beneficial. And that's why those treatments persisted for so long, in spite of their harmful direct effects. So things are not necessarily better today in many respects. This is a meta-analysis from Wayne Jones and colleagues of surgery across several different conditions, so pain, obesity, gastric reflux, essentially, and other conditions. And what you can see here is treatments that are pretty large, but it wasn't until these things were widely used in clinical practice, Medicare covered and insurance reimbursed, that people started to go back and actually do randomized controlled trials to test whether they're effective. How do you do that? You give a sham surgery. So you don't do the full surgery, and so on. And here you find that with the sham surgery, the responses are sometimes comparable or nearly as large as the actual. So a lot of the benefit of surgery is just the context and care itself. One of the first people to realize this was Bruce Moseley, who is a physician in Texas and happens to be the team physician for the Houston Rockets, or at least he was at the time, and he started to suspect that the knee surgeries that they were doing were actually, in large part, placebo. And so they did the first randomized controlled trial, first an open label study in which a smaller number of patients really got better, and then a larger randomized controlled trial which showed no benefit of the surgery over sham surgery. And I'm going to show this little video. I have to be tricky here because I'm going to show you the audio from my computer. Hopefully it'll sync up. But this is from a BBS documentary, and so I'll let Bruce tell you a little bit about the surgery. And then you're also going to hear a little clip I cut in from one of the patients that they interviewed several years after getting the sham surgery. And you'll hear what the pain was like before, and then something about after the placebo surgery.
You should know
that. He does now, right? They know now, but at the time he didn't know until after the procedure.
We had a very elaborate ruse, if you will. I would play a videotape on the TV of a real surgery. While I was watching the TV, I would ask for instruments and pass things back and forth and carry on conversation as if it was what I would really have done if what was happening on the TV screen was actually going on during that surgery.
Can you describe what the pain was like before the operation?
Terrible, awful. I didn't think that it could be any worse. After that,
After the surgery.
I was able to do that. He could. Woo! We could go dancing. He took me dancing, and he could dance all night.
I went from being a skeptic to a believer. I realized that we were really onto something. There are clearly cases where we do surgeries, and it's as much or more the patient's feeling about the surgery that influences the results as the actual surgery itself.
And this is not limited to arthroscopic knee surgery. There are a number of other widely used reimbursable procedures that you can pay good money for in the U.S. healthcare system that also don't beat placebo effects in clinical trials and meta-analyses, including vertebroplasty, which is for osteoporotic fractures, epidural steroid injections for back pain, which many people in my family have experienced even recently. And this really surprised me, stents for angina or chest pain. It doesn't mean that stents are not important for cardiovascular disease at all, but in this paper, which was one of the top medical journals in the world, there was no benefit or aversion for this outcome. Such failures that are attributable in part to large placebo effects also apply to other emerging treatments that many of us are very hopeful about, including dopamine therapy for Parkinson's disease, gene therapy, and a fairly famous study recently, which is a deep brain stimulation for depression and the ventromedial prefrontal cortex stimulation. In this study, there were these open-label trials with, again, famous physicians doing treatments, and a lot of hope because people with refractory depression, 2/3 of them were getting better. For the first time in years, waking up and feeling better. They did the RCT with a sham surgery, multi-center industry-funded trial. The response rate was, I think, 23%, give or take. And in the treatment group, this is pretty good considering it's very depression. But in the placebo group, 18%, no significant difference. They stopped the trial early after that. So placebo effects are also growing across time in several domains, including pain. But drug effects are not, at least not in the U.S. in pain trials. And that means that the drug-placebo difference is shrinking. And this is, unclear what's causing this exactly, but one of the potential causes is that in the U.S., there is direct-to-consumer marketing. So patients know what drugs they want to get, which are new drugs. And that might have an impact. And also, those trials are getting longer and more expensive, more procedures and more follow-up. And so the placebo component might actually be growing. Trials. So I picked out this example because this really surprised me. This is an industry-funded study of a gene therapy for Parkinson's disease, that is injected into the striatum with dopamine-related areas in human beings to modify their genes to increase dopamine efficacy. And monkeys in Science and Nature had shown beneficial effects. The company had an earlier failed trial where they put all their eggs into this basket that it said, the early trial was too short. We're going to follow people up for 2 years and really nail this and then go to market. So this is motor symptom improvement. You see here on the y-axis. And you can see here patients drop. This is after 6 months. And they stay better for about 2 years. Unfortunately, this trial failed. And it was caused the drug company to be sold off for parts. Why? People improved in the placebo group and stayed better for 2 years. And to me, this is crazy. Because this Parkinson's is a neurodegenerative disorder. You don't just get better. Why? And it was also industry funded, which means that practice is significantly more likely to show a drug versus placebo benefit. Okay. We've been studying placebo effects for years, and we think that placebo effects provide a window into the treatment context, which is actually multifaceted. So I don't think that giving people fake drugs is a way to actually make them better, but it is a way to study what happens in a supportive caring context in various ways. So there are various aspects of the external context, like by a authoritative, warm, caring physician or other expert associated with eye gaze and body language, voice cues, and so forth. Treatment cues, like the syringe or needle or other things like that. And then a rich internal context, which involves expectations about pain. And what I think is important is meaning schemas. So changing the narrative of what you tell yourself about pain, replace what it means going forward to you. And this is a very complex family of there are also precognitive or preconceptual processes that in certain cases actually make a big difference. So think lower level forms of learning induced by cognition. Many aspects of the treatment context can be tapped into clinically, even without deceiving people or giving people a placebo. And there's another aspect of pain in particular, which is really important, because in our medical system and even worldwide, pain is often invisible. You can hear people's reports of pain, but you can't see it often. It's not caused by an obvious lesion. In fact, in some cases, you need an obvious lesion or deformation in order to have a legal case, for example. Otherwise, nice. So many people are ignored when they come with real complaints of pain, and it's complicated to figure it out. So this is a woman who is writing this, and she's like, I can't count how many physicians I went to. They told me nobody can have that many things wrong with them, because she had hysterectomy surgery and she stayed on the couch for 6 months, and she had headaches and other kinds of headaches and autoimmune problems and so forth. So what happens is people like this just get labeled as complainers. One physician asked her, "What's wrong with you? Why are you seeking all this attention?" And this happens to be Jan Chambers, who's the president of the National Fibromyalgia and Chronic Pain Association. So I heard her story at a meeting in Washington, and she's published this on her blog. So it's really a surprising story that is recapitulated again and again, including, I have to say, by the former dean of Stanford Medical School, who also had a similar experience, where when he had an injury that caused chronic pain, people started wondering if he was really just depressed or unhappy with his job. Another aspect of the roots of this goes way back to at least to the mesmerism. So Franz Mesmer famously healed people essentially with magnets. This is the subject of possibly the first ever study in psychology, which debunked this idea that the magnets were having an active effect. And Ben Franklin was one of the people here who was on this committee, along with Lavoisier, the chemist, and other people. And they concluded after a series of experiments, essentially, that these are real changes that are happening with people, but they're produced by the people's imagination. And so, so Mesmer was debunked. But some followers of Mesmer, like Desmond, pointed this out, which is interesting to think about. He said, well, if the imagination is so powerful, it can really help us be well or ill, then wouldn't that be really helpful to tap into? And so, I sort of agree. And we've had an ongoing debate about, whether placebo effects and context effects and even psychotherapy can actually be effective or not in an real context. So here, there are competing headlines. This is from the late 90s. Placebos proved so powerful, even experts are surprised, they cure your knee pain, etc. But a few years later, competing science headlines, placebo effects are more myth than science. After this meta-analysis was published, where sort of ironically, they found placebo effects in pain and in some other areas like obesity, continued to do so in subsequent meta-analyses, but they attributed those benefits in pain reports and clinical outcomes to people just sort of like, main characteristics, saying they feel better. So it's a real question. So when we started this research, this gave us a good reason to turn to the brain. So there's a real debate about which effects count. So if there's a pathophysiological mechanism, that might for a treatment, then it gets treated differently by the FDA and by other approvals and by the court system and so on. Even though what the FDA really cares about is healing, functionality, and survival. So we prioritize physiological mechanisms. So I figured, like 15 years ago, I guess, that if we could really kind of move forward with trying to find evidence for physiological correlates of pain in the brain, that would help us get a handle on whether treatments like placebo and context effects have important or relevant brain interventions. Okay, so this is a range of different placebo treatments that we give to people, injections, pills, nasal sprays, creams, and others. And this is a summary of version 1.0 of placebo research. There are reliable activity decreases across studies. So you see each blue dot is a study showing activity decreases during pain. In some cases, there are causal effects of the placebo in randomized trials. And then the lighter circles are correlations. And activity increases in other areas. And by and large, those activity decreases look approximately like they are serotonin systems that convey important information about pain. Somatosensory cortex, anterior cingulate, anterior insula. And the increases are largely in areas that I'll talk a little bit more about. But for now, I'll just say I think that they're involved in the construction of situational meaning, situation representation. There's a little different talk I would give that explores that. And so that literature as a whole has helped to change our views of what pain and other symptoms are. So at first we thought of pain simply as a function of ascending nociceptive input, reaches the brain, and the brain magically just sort of conveys the pain experience. And then version 2 was the brain had an off switch for pain, descending inhibitory system. And then it had off and on switches. And now we've moved from that kind of view to thinking about what the frontal cortex and other sort of higher level brain systems are doing as providing rich representations of the context. What's happening and what should I feel right now? And that what should I feel right now doesn't have to be conscious, but sense projections back that influence all of these descending areas and provide contextual control over the signal. So if you think about it this way, then pain and other symptoms are inferences made by the brain that are adaptive. And they're a function of both sensory evidence and of context-based priors. And those inferred representations can take multiple forms. It can change how you feel, your experience and reported experience. It can change your autonomic physiology. It can change the course of learning itself. And so for most of this talk, I'm going to focus in on the learning bit, because I think that's particularly important. And we've moved, again, from thinking of the VMPS unit as being a kind of a volume knob or value switch or whatever it is, towards thinking of this as a representational space, where there's a rich representation of multiple elements, including how those elements relate to oneself. And the closer something is to yourself, then the more emotional it is, and the more it generates physiological responses that are emotional. And for this reason, I think that the VMPSE is, broadly speaking, part of all forms of psychopathology that I know of. The idea is if a placebo treatment or any other psychosocial intervention can change the learning process in the brain, that's important because you can do an intervention at one time point, and that can change the trajectory of what people take away from the experience for who knows, the rest of their lives, maybe. So, this is maybe, how do you get a placebo effect in Parkinson's the last 2 years? So this is the first study that we did to try to get at this just a little bit. And this is a study of Parkinson's patients, 20 or so patients. And they all brought in their daily medication, which we then crushed up and dissolved in orange juice in front of them. And we said, okay, we're going to scan your brain when you're, before drinking the orange juice and after drinking the orange juice. So off drug, on drug. And so in fact, we have this group here. They get scanned first off, and then they drink the orange juice, but it's not the real orange juice. Don't tell. It's actually, a different glass of orange juice with some bitters in it. So no drug. The other group got the reverse order, the control for time. So for within-person crossover design. Afterwards, we gave everybody the real drug and test them again to see how it would compare. We looked at 2 tasks, reward learning tasks and motor learning tasks. I'm going to talk about the reward learning task. And this is the structure of the task. It's fairly simple. You do 2 symbols, call them A and B, and you make a choice. And then you either get rewarded or you don't. Your job is to learn over time which symbols are rewarded most frequently. There's also a punishment learning task. And my postdoc, Leanne Schmidt, who did this, they call this task, this came from Matthias Castiglione, who's now in Paris, and they call this the mother task because it's been used lots of different times in context. It's sensitive to PARC and computation status and dopamine loss. It's also sensitive to dopamine repletion with L-DOPA in healthy patients. So that's kind of why we picked this. Okay, so this is what happens with the learning performance. So this is learning from reward, and this is off drug. They learn fairly slowly. On placebo, they learn significantly faster. And on drug, they learn just as fast as on the placebo. So here, the placebo is just as effective as drug in this case. Learning from punishment, slow, and there's no differences across groups. So then we can look at the brain and construct a regressor over time, which we're going to correlate with brain activity. And this regressor can take the form of trial-to-trial variation in expected value. So what happens in this simple learning circuit is, and this is a sort of theory, but it's sketched out, is when you get some good outcome, it's compared with what you expected, and the expectations are maintained in part in the VNC. Comparison process takes place in part in the VTA, ventral tegmental area, where subset of dopamine neurons fire in proportion to prediction errors, whether something is better than expected. And if something is better than expected, then you update the expectation. Choose that thing more often next time. So this is, we can look at correlates of expectations and prediction errors. And in a nutshell, off drug, we don't see very much in terms of correlations. On drug, we see a significant correlation between BMP and placebo and expected value. And on placebo, we see something similar. The critical comparison is the direct comparison, placebo versus no treatment. And here we see significant difference in that area. So there's a placebo benefit in essentially, we think, the representation of expected value signal. We saw similar things in the incumbents for prediction error, but I'm skipping over some stuff for time. Many ambitions to tell you lots of stuff. So, if we turn to pain, how does context shape pain processing experience? There are several different options. One option is if you have some stimulus, some system that transduce or create or construct the experience of pain, and then the pain report the outcome. These context systems might work in at least two different ways. They might work by modulating the sensory input, which clearly happens in certain cases. But there are questions about how large those effects are, and they can happen in ways that are independent of pain systems by modulating the precision value and other aspects of the pain beyond these sort of more immediate nociceptive systems. I'll call those sensory modification and behavior modification. And both of those things can interact with learning processes. So over time, those effects can be strengthened. Okay. I think this depends on the particular context in which one finds oneself. And I think that there's a lot of animal work on descending modulatory systems. It clearly can happen, but very little work on human intervention. I think the key questions are, when do you get such effects? How big are those effects? What makes them persist over time versus not? Okay. So now I'm going to take a small detour into how do we measure pain in the brain anyway? I'm going to come back to this at the end and apply this to the study of placebo. So when we do studies like placebo studies or others, what we're looking for is a biomarker in the brain. And a biomarker is, what we actually really want is a representation, which is really an information structure. The idea that if we had some physical brain pattern that if we stimulated it, we could recapitulate pain every time. And if we blocked it, we'd never feel pain no matter what, right? We can't, I think that's actually an abstract concept. We can't get at that. But one step toward it is to define a biomarker that's a measured pattern here of brain activity that responds to noxious events and predicts experiences or other outcomes that we care about. And at a minimum, a biomarker ought to be sensitive and specific to the process that it's intended to measure. And for me, when I started doing neuroimaging a little while back now as a graduate student, we just never thought about brain measures in this way. And it was really only in, I would say, the last 5 or 6 years that we have sort of started treating brain patterns as you would treat any other measure, and evaluating their sensitivity and specificity and generalizability across studies and contexts, and validating them in that way. And so that's guided a lot of what we do in the lab. I think it's an exciting direction. So such biomarkers are gateways to measures of reference. And traditional brain mapping approaches have a number of problems. One of the main problems is a problem of inference. And so I like to say any neuroimager worth their salt ought to be able to look at this map and say, well, what task would be performed? So you can take a guess if you want. So if you've seen my talk before, don't take a guess. You don't have to guess, I'll help you. So there's the anterior cingulate cortex, which is a nociceptive target, so pain-related inflammation, pain-related neurons. There's the anterior and mid-insula, also pain-related, medial thalamus, major target of ascending pain pathways, somatosensory cortex, S1 and S2, which turns out are fairly selectively activated by pain and touch. So that's a clue. We can do a little better. We can put this into a database called neuroscientific.org, which was created by a postdoc, Cal Yarconi, in my lab, who's continued to take this and develop it, and now has activation coordinates from over 14,000 studies, and it also has the full text of all the studies. So we can plug in this map, which I did, and say, well, things that activate this, what do they tend to talk about in the paper? And they talk about noxious heat, sensory painful, sensation, stimulation, muscle, temperature. So that's a stronger clue that this is a pain-related map. But unfortunately, this is not pain-related at all. This is a map of what happens when you look at the person who rejected you in love. Similar, many of the same areas, but not the same thing. So in the old days, when we wanted to test placebo effects, I'll pick on our study, you find things that respond when you turn up the pain, the heat, right? And these are such things, they're all very sensible. And then you say, okay, well, are they reduced with the placebo? And here they are. Here's the control, and here's the placebo. This is, I think, the insula. And so you can see a reduction. But, you know, that's great, but, you know, it's not entirely satisfying. And this is why. When we start to look at any given part of the anterior cingulate cortex, like this, which is one of the most pain-selective parts of the anterior cingulate cortex, and look at all the different things that activate it across studies, what we find is kind of all over the place, right? Language, attention, motor, memory, social cognition, language, reward. So you know, when I'm in this, so the positive predictive value for pain is very low. It's clearly pain-related. There are nociceptive neurons in there, right? You can't just say anterior cingulate, that must be pain. So in fact, that placebo effect might have affected pain construction systems, or it might have affected something else, like how you describe your pain, or how you think about your pain, or how you feel emotionally about your pain. It's all very sensible. So we really need better targeting. Empirical question, how close we can get, which kinds of outcomes to useful measures. Now, the idea that we've shifted to, the idea of creating strong predictive models, which are precisely defined so we can validate or invalidate. And it's not that I have physics envy, but this is pretty cool. So when Albert Einstein first developed this theory of relativity, he did not become famous overnight. He actually was still an obscure theoretical physicist, and it wasn't until several years later, I think in 1919, if I remember right, that Eddington, this eclipse happened, and Eddington tested this by measuring the bending of starlight around the sun. And then you could see for the first time the blending effect, and then Einstein became a sensation overnight. And it's not as though, you know, I think that we need to recapitulate physics in every way, but I think what's important from this is the idea that that there was a strong predictive model that was tested and can be empirically validated, even if that comes later. But my hope is to treat the brain maps the same way. And most of what we've been doing is, in fact, creating maps, where you look at different little bits of the brain and say, what's happening here, what's happening here, what's happening here, so this responds to pain. But that doesn't really explain much about how pain is constructed. What we really need is models. So in this model, so the Earth goes around the sun and the moon goes around the Earth. And at least at a conceptual level, it's a much simpler problem, right? Ellipsoid around an ellipsoid, rather than a sort of very complex, obscure set of differential equations. So models can be simplified. And the brain patterns that I'm talking about now are really probably the simplest possible prototype. So the idea is that, given a set of brain measures, we want to make a reverse inference about the mental state. So we have a set of brain predictors that we take as a whole, and those can be linked to different times. We like those links to be as tight as possible, and then we can test them and do studies. So when we turn back to the dorsal anterior cingulate cortex, which is a overall non-specific area of the brain, we can really start to pull it apart, and we can ask this question. So it's activated by lots of things. That might be because there's one underlying factor, there's one thing that the cingulate does that causes all these activations, like expected value of control or other similar theories. And there are many such theories that have been proposed. On the other hand, the anterior cingulate cortex, every voxel contains approximately 5.5 million neurons, and they do different things. So if you go to the neuroscience literature, you find that there are separate neural ensembles for different kinds of motivational functions, including all of these here, and even some subcategories. So there might be something that conveys a sense of pain that's not shared, and just we're mixing the signals all together. And there's hope that we might be able to pull this apart. So this is a simulation that I created with two types of intermixed neurons. We'll just call them type 1 and type 2. If you take these neurons, they're pretty homogeneously intermixed. In fact, it's a homogeneous Gaussian process, and or Poisson process, right? But and we can sample those things on voxels, which are kind of coarse, and approximate the local field potential by kind of smoothing them. There's type 1, there's type 2, and then when we get these voxel patterns, they're over here. So they're a poor representation of the underlying neuronal signal. But what you can see is, even though any one given voxel is a mixture of both of those neuronal types, and doesn't have very much signal, across the patterns look quite different. So that's part of the hope, right, that we can differentiate at the pattern level. It doesn't mean this is always going to work, but I think in some cases it really can. So we, in this study I'll tell you about now, we kind of really pushed this forward and took a construct validation approach, going back to the work by Paul Mila and others. And the idea is, if you have a construct like pain, then you ought to try to devise multiple measures of that are as dissimilar as possible, to essentially triangulate, right? So there might be different kinds of pain, thermomechanical and visceral. And you might have a second construct, like emotion-related construct. There might be several different kinds of emotion, social pain, emotional pictures, and emotional sounds in this case. And then you can validate measures of each of those, and then you can finally ask how are they related, how they're working. Okay, so if there are two separable constructs, you'll see this pattern of intercorrelation within measure. If there's one big construct and everything's kind of intercorrelated, you'll see something like this. And no reliable constructs, every measure is just different. So this is the logic that we built on in this study, where we're measuring multiple indicators of each domain. So in this case, we have 270 subjects total, and they're split into 3 kinds of pain. And there's 2 studies of each kind, 15 subjects each study. And then there's 3 kinds of cognitive control. So this is working memory, response selection, and response competition. So 3 different tasks, and 3 different kinds of negative emotion, elicited by both pictures and sounds that I just went over. And so then what we can do is take the correlation matrix in the anterior cingulate cortex across all of the subjects. So this is 270 by 270, similarity of all these brain maps. And we can decompose that correlation matrix into parts that we care about. So we can model that matrix. And so here you can see this pain-related thing that I showed you here. These are study-specific effects if these are strong. These generalize across subdomains or 2 different types of pain, let's say, or more. And this part of the matrix is generalizable across the 3 kinds of. There's also relationships across them. So I won't go into details about the modeling, but we did a lot of work to kind of validate the model, and then it works right. in this case. And here we can then pull out whether there is, in fact, an appreciable correlation here, which would be a domain general representation of pain or emotion or cognitive control. I'll just turn to the cingulate cortex, although there's lots of results from other areas. And what we can see here is across the three kinds of pain, there clearly are three distinct kinds of patterns. They don't look the same. And only with pain is there evidence for a generalizable representation across the multiple. It doesn't mean that these other tasks don't activate that same bit of cortex at all, right? It just means that the patterns they evoke are A, not the same as pain, and B, they're not internally coherent across different types of cognitive control. So it makes sense. Different tasks, different patterns of activity. Okay.
So can I ask one?
Yeah,
One
of course.
question about the method. Are you doing the similarity matrices on individual subjects?
Yeah, each of those is individual subjects. So 270 subjects with that 270 by 270 patterns. So I think, if we step back for a second, the idea of defining a useful measure of pain or any other outcome is a challenging thing. We have to do it across studies. And we've been working on with, several different groups about sort of exploring what would this take, what kinds of criteria should we apply to any measure. This doesn't have to just be brain imaging. It could be EEG, or it could even be that diagnostic test that you got at a clinic last week, right, in a blood pressure. For here, with biomarkers, what we like is precisely defined signatures or patterns, models that we can replicate, and they're not fuzzy, they don't move over time, right? They have to be replicated and applied without adjustment across laboratories. So there has to be a method for taking somebody else's model, plugging in some data, and then making a strong prediction. Validating yes or no. How will it work? Right? And it should be sensitive and specific to the kind of outcome that you're interested in. So when we go to pain, measures of pain using fMRI, the idea is we can identify a potential signature pattern by taking a training sample and taking each person 4 levels of painful heat here, and then training to minimize the error on predicting pain reports. This is a linear model in this case, so we get out of it a signature pattern, which is the best set of weights that are the most predictive. We call this pattern the neurologic pain signature because our journal editors required that we call it that, and we didn't want to mess it up. It is a precisely specified pattern within the class areas, and those areas are sensible. Many of them are, in fact, targets of pain. So it's not that you have activity anywhere in the anterior cingulate cortex. Important. What's important is that you have that particular pattern, right, and you test that, so it doesn't move around. We think that pattern is important. So then we can take new test data. We take people 1 person at a time, and we can apply this signature pattern to their brain images. And that just means we essentially take a weighted average where the signature supplies the weights. And then we turn a complex brain image into a number, which is the predicted, in this case, or the pattern response. We can validate that. So this is an application to a new cohort, a new scanner, and a different sample. You can see that the lines go up over time. Each person's a line. And so in general, increasing heat, painful heat, it goes up. And we can take that continuous readout and turn it into a classifier. If we had to guess which is more painful, high versus low simulation in this case, without knowing the answer ahead of time, we'd get the answer right 96% of the time. And that's been our benchmark value approximately across studies. We've been testing its sensitivity and specificity in a lot of ways. I'll show you one of the validation studies, which comes from a meta-analysis of pain and placebo effects. So in this study, this is a collection, a collaboration with multiple groups you see here around the world, many of them in Europe, 20 different studies, and each of these is a study here. And what you can see is the shift to the right for each study means that this pain signature responds to painful stimulation positively in the meta-analysis. And each of those dots is the individual participant. What you can see is, across 600 people and 20 sites, this pattern responds robustly to multiple types of both experimental pain. There are really large effects. Effect size is greater than 2. Over 95% of the individuals show that response. So that's a sort of good sort of first pass, validation. This is pain related. We can also validate its specificity. And I won't show you all the details of this, of course, but we've been sharing data with sites around the world, and anybody who has something that, they think, well, this looks a lot like pain, but it's not pain. Maybe it's not being able to breathe, or maybe it's something else, right? So we can test this much faster than we would if we had to run all these studies de novo. And this is a quick snapshot of the profile of sensitivity and specificity. So it's activated by or sensitive to things that are multiple types of noxious or painful stimulation, including things that we wouldn't test in our lab personally, like gastric, rectal, esophageal distension. People drop in Europe, it's dropping balloons down into people's stomachs, inflating them, stuff like that, right? And including some responses, for breathlessness and aversive taste, although it doesn't scale with the intensity. It doesn't respond, or it does respond to many kinds of treatments, including opioid treatment, SSRIs, and other kinds of foods. I'm going to come back to this in a little bit. And it has a profile of specificity that I think is really promising. So, how do we test specificity? Well, it's difficult to match pain on every possible thing that it could be matched with. And so there are open questions about what this pattern is really measuring. It could be something that's very specific to some types of pain, multiple types of pain. It could be anything that's negative or arousing. And so what we like to do is find something that's also aversive, arousing, salient, but not pain, and test that. So I'll go back to romantic rejection, which is one of the experiences that often happens in life. The experiences can last a lifetime. This is a man who's proposing to his girlfriend at what I think is a Knicks game. And as you'll see, it's not going to go well. All right. So yeah, good thing I'm already married. All right, so we took 60 people in the study. They all were recipients of an unwanted breakup. They viewed pictures of ex-partners and friends and experienced painful events. So just to pick on myself, this would be my friend, and then my ex-partner, and then I would experience both conditions. Okay. And they're very similar in terms of negative affect ratings and brain activity. So what about the brain? Well, we can validate that it responds to hot versus warm stimuli. What about the rejector? We don't see any. And when we look at the pattern, we see it really responds when it's actually painful. There's a threshold for pain, no pain classification, and then there's no response. So that's one of the ways in which we can validate that. And we can see in the anterior cingulate cortex as well, there are different patterns of activity within the same regions that indeed code for pain versus other things. So there's a pain-related pattern in the anterior cingulate cortex that responds only to pain, but not the partner. And likewise, the same set of voxels, a different pattern that responds only to the partner. And these things are uncorrelated. Okay, so that's just a drop about specificity. And it's not activated by a number of things that are often thought to be confusable. Okay, I'm going to skip a little bit. I'm sorry, I'm running a little bit behind a lot. So I'm going to skip it. But now we can turn back to the study of placebo. And we can ask, well, if we take the NPS as a proxy, how, in what conditions can we see evidence for this kind of early life? When you look at placebo effects, standard placebo effects, which you just validated with pain, this is what we see in behavior. So almost every study shows a reduction with placebo treatment versus control. That's good. And what about the brain? There's the generalization, and here's the brain. Very little response. There's a significant effect. The effect size is very small. It is correlated with the behavioral analgesia, but it's just not large enough to explain what's happening. So this points to contributions from other systems. There's a number of things we can do with this from here. Not all placebo effects are created equal. So we can look at which interventions modulate this system and when and under what conditions. We can essentially use this as a tool for treatment design. So that's what I'll just show you the drop on here. and then I'm going to wrap up. So which psychological treatments work on the NPS? You can start to compare data across our studies and other studies. And now here what you see is each, what do you call that? Position on the x-axis here is a study. Each dot's a subject. And if there's a positive bar, that's going to mean an effect in the expected direction. So we can look at some placebo studies, and we don't see any response either. Or studies where reward just precedes pain. Yeah, okay, so I'm okay. Yeah. Only time for questions.
Yeah.
But I shouldn't feel stressed.
Yeah.
All right, I'm going to relax. Thank you. Yeah.
You're relaxing. That's a clarifying question on your previous slide. So you said the neurological, the NPS doesn't show placebo, but it correlates with the behavioral reports pain reduction?
It does to some degree, and the correlation is modest. I can't remember what you.
So it's like the brain is responding, but
Yeah, there's some evidence in some subjects some of the time, this is probably happening, right? But overall, it's not explaining the behavioral phenomenon. Sometimes so dramatic. Yeah. Thank you. And so, reward right before pain, all of these things, by the way, produce reported pain relief. There's no effect of this. Negative images during pain, analgesics, but no effect of the signature. Perceived control, something I would have been very interested in, hopeful about, no effect. Reappraisal of pain, rethinking pain up and down, right, to make it better or worse. No effect. But that doesn't mean that no psychological interventions have effects. Here is one example of things that do. I'll show you a drop of what this is in a second, but we call it, reinforced expectation. Giving people the right kind of expectation at the start, reinforcing it with experience under the right conditions, can in fact create significant effects there in 4 studies. And then here's one study of an opiate drug, and a study of positive images, which in this case were erotic images shown to men, which also has an effect, something to follow up on later. So I'm going to unpack this. And actually, so since making this slide, in recent studies, we found a series of other things that also work that are modulations of social contact, like holding the hand of your romantic partner while they're experiencing, or accepting pain on behalf of the partner, to take away their pain so you voluntarily take pain. So these kinds of things, and also meditation like states can also happen. That'll come in the next version. But let me unpack this reinforced expectation. Yeah. And this is a kind of paradigm that we found is quite powerful. So here it's a conditioning paradigm. So there are low Q trials and high Q trials. But in this case, we don't reinforce those with actual stimuli. What we do is, you can't see this, we reinforce them with pictures of thermometers that are either low or high. So they experience this symbolic association, right, with what's going to happen later, but they don't give you anything. So there's no low-level conditioning. Then in phase 2, there's a pain test where they get low Q and high Q trials and neutral Q. Here, the levels of heat that we give people are identical. So the only difference is in the Q values. And these Qs are never reinforced, so they should cause extinction. Right? So the distribution of heat following these Qs mean nothing, right? So according to learning theory, they should go away over time. So what happens? Well, this is what happens with the low Q across 2 different heat intensities. And here is the high Q, substantially higher values for pain ratings. We can also look at autonomic responses. So that's with the low Q. And with the high Q, substantially higher autonomic. A curious thing that we find is when we look at this over time, these effects do not extinguish. So those are expectation ratings for high and low, and here are the pain reports for high and low. Now here's a replication study doing the same thing. So people are telling us they still believe that the high Q is high and the low Q is low across time. And there's still a benefit in pain reports. So there's no extinction. And this, to me, harkens back to the Parkinson's study a little bit that I showed you before. We have these long-lasting effects. And so we here wanted to explore how this could come about. How can you create this sort of positive extinction-resistant feedback cycle? And indeed, if expectations influence experience, and experience influences expectations, and that experience is also essentially the teaching signal that drives future, then you can end up with a self-fulfilling process. So in this study, we were able to model this because we independently manipulated both the stimulus intensity here and the cues as instrumental variables. And so what we can see is across both study 1 and study 2, these are quintiles controlling for the cue type here. This is study 1, this is study 2. That the greater the expectation of pain, then the greater the pain experience reported in that trial in both studies. And conversely, here, this other limb where the greater the pain, then the greater the update in expectation. So those are the ingredients of this dynamic. And indeed, following along with that, if we actually look at the brain responses to high versus low cues, these are really robust differences across the brain. We can look at our NPS signature measure and see, yes, in fact, it responds to the intensity of the stimulus, but it's also higher for the high cue than the low cue. So this unpacks one of those bars that you saw. So here, for me, this is interesting because this is an example of cognitive expectations without any primary reinforcement. No low location in nociceptive systems. What there is a conceptual association that this thing means that I'm going to experience higher pain. So that is one part of the mechanism. And I'll be brief about this, but a lot of what we did in this paper is actually unpack the second. So it turns out that you can show that if you dampen experience in proportion to expectations, right? Expect less pain, I get less pain. That can slow extinction, but it can't create a self-fulfilling prophecy. And if it did, we'd be hallucinating stuff all the time, right? I mean, it would be crazy. You know, you expect pain and it hurts, and your sensory system doesn't work properly. We think that effect is actually relatively small. But the second effect is bigger. And this is an effect that's a confirmation bias in learning. And so we modeled this in a couple ways, and I'll show you kind of the outcome of what the model looks like. Explain it. So here, the first level, there's pain processing. So it's input, reinforcement, and then the pain response. Okay, and that's controlled by this gamma parameter here. And the level 2 is this association learning level, where you get a cue, and that cue leads to an expectation. And that expectation is compared with what you got. That generates a prediction error. And when that prediction error is worse than expected, you update that association and say, hey, this cue is pretty bad. Right, so that's the learning effect. Now, the first pain modulation effect is you get the pain you expect. So that's gamma here. So expectation reduces that reinforcement. And that's part of the story. But the second part of the story is as follows. Third level is our beliefs about what's happening govern what we learn from experience, which is essentially the main theme of this talk. And here, we model this with two different learning weights. There's a confirmatory learning weight when the cue matches or the experience matches your initial expectation, and a disconfirming when it doesn't. And so essentially, and this was a significant, both of these things are significant in those studies. It's about 2/3 confirmation bias and 1/3 pain manipulation. And so essentially what's happening is, if you expected the thing to be high pain cue, and you've got something that's even higher, it's really bad, you go, yep, that's it, right? And you update, you learn. But if it's less painful than you expected, you go, maybe it was a fluke. That was a, who knows? I'm not sure what I felt. You discount the experience. And that's kind of what we think is happening at the heuristic level. So we modeled it this way with a computational reinforcement learning model, and we also modeled it with the hierarchical Bayesian model, and you get the same answers both ways. Both of these parameters are really critical. I like to see things in the data. I like models, but I don't always trust them. A lot of assumptions. And so this is a sort of more empirical view, where what you can see here is the empirical learning rate, which is the expectation update from trial to trial, following different types of experiences. And here, what I've done is I've just put stars over all of the violin plots here, where the experience confirmed the initial expectation, or the initial value of the cue. Right? So if you got, a low pain cue here, and you got a lower than expected experience, you update a lot. If you got a high pain cue and it's a higher than expected experience, you update a lot. That's true for both studies. So that's another kind of evidence for confirmation. So I think this is one step towards, trying to capture how this process might unfold over time, how a brief intervention might play out in lasting changes. So I've told you an awful lot, very ambitious attempt here. I've talked to you about the importance of beliefs and their prevalence throughout history. I've talked about placebo effects on pain and symptoms and learning, with an emphasis on the learning processes and how to really unpack what's going on. We should combine interventions in computational models with biomarkers that are developed to track particular aspects of processes. And that's been a very fruitful direction for us. I talked about a biomarker for pain, it's a provisional one. Some about the generalizability of that and the construct validation process that underlies it. And then how we can use that to both test placebo effects and learn something new that, the standard placebo paradigm is not as powerful as I had thought in really affecting pain-related behavior. Doesn't mean it's not important, in the world and in behavior. But I learned something new here. And then finally, we can use this kind of measure to refine our intervention and understand which things affect particular brain systems. Okay, with that, I'll leave you with the final thought, which is this view, which I've really come to believe in, of symptoms as emergent properties, as adaptive inferences that are based on our prior beliefs, conscious and unconscious. And that determines what we take away from the experience, and therefore what we do. I'm also going to say thank you to my collaborators and lab members, many people who worked on this, and to all the people who contributed data so far. Some of them are not on these slides, probably yet, but it's been a really fun process of working with multiple groups to test and develop these. And I'll also say thank you for your
So this is sort of on the methodological side, but if I understand when you did the RSA, you were sort of pulling apart different kinds of pain, right? Is that right?
Yeah, or we're trying to generalize across different kinds of pain. Right? So the thing is, is there a common pattern that underlies the three kinds, right? Across studies.
And that pattern was really at the individual level. But it's ultimately,
sorry that wasn't clear. It's at the group level because the input data was a series of maps of pain versus high pain versus low pain, right? For each person in those studies. So something was at 90 people with high pain versus low pain. So the pattern that comes out is yes, it's a group pattern. So we're losing stuff that's happening, person to person idiosyncrasies, and the pattern is reflecting the sort of average across. The part of the pattern that's consistent with the pain, right? And by the way, the patterns that you see were trained using a Hartland squared model. So I didn't talk about that, but yeah.
So sorry, now I'm slipping a little bit. So now are you talking about the machine learning and the neural signature, right?
No, sorry, I was talking about that. Yeah, so there's the RSA, right? So there's that. And basically, we made inferences about constructs, right? Used by decomposing the correlation matrix. Right. So that gives us statistical inference. But when you want to visualize those patterns I showed you, that's the other. Right. To say, okay, show me the pattern across voxels that best tracks the pain across.
We try to deal with the research very interesting. Just wondering, what we design, in intervention, like including educate the patient. So what's your suggestion? How do you use your results, like how the patient, there's a high pain or low pain, so how do you use your study in design intervention?
Yeah, one of the I have a lot of thoughts about that, and I'd love to have a conversation, but a sort of quick answer to that is, if you kind of take the implications of all these things seriously, then a lot of what helps a patient either get better or not is going to be their core beliefs or their narrative about what caused their pain in the first place, and what it means to move and do things that might hurt or so forth. And so, again, long conversation, but we've just run a back pain study, which is a treatment study that employs these principles. So we're still working on that. It would be a different talk, but also it borrows from things that a lot of people are doing, aspects of ACT and CBT and physiotherapy and so on. And I think the principles are what we did and what I see people doing that's effective is, first, I think people need to know that they can get better. Sometimes people are told that you're always going to be in pain. It's always, you can manage it, but and so there's got to be a sort of a commitment to be able to say, yeah, you can actually really improve. The change in the narrative is, in some cases, it's the pain is actually avoidance learning system. It's not in their back of their knee or their neck. And so that change in narrative, so our therapist was telling people, look, you can move, you can do things that are painful because the pain is actually caused by brain changes, and you can unlearn those by attending to your pain, exploring your pain, doing things that hurt. And so then there's a shift right from sort of avoidance and fear towards active approach of that pain sensation, but in a way that's not sort of threatening, right? So that's I don't know if that came through at all, but that's but I've seen like various pieces of evidence from different therapies that seem pretty amazing. Like when I went to Belgium, they tell yeah, this is what we do. We do pain exposure therapy, and that's the first line of thing that they do is try to convince somebody that something that they thought was disabled is actually something that they can do safely and do it. I'd love to hear more about how that fits with your approach.
So surely more out of my studies is looking at therapy and how people, how it changes the biophysical, whatever symptoms that happen. And so I'd be interested in seeing if the next steps for you in this program. Are you looking to possibly using it for how to optimize where you're going to target for the intervention? I mean,
right.
What you're saying, it's almost like matchmaking could be intervention for somebody, like,
Yeah. I mean, yeah, so I think that the story, I think, about how this interfaces with clinical stuff kind of goes beyond this signature. So what we're finding in a nutshell is that this NPS seems to be really darn good for evoked pain. But people with ongoing clinical pain, there are a number of other changes independent of that, potentially, that are important. So one thing we're trying to do is, validate and test evoked pain. So we tested fibromyalgia, knee osteoarthritis, and some treatment, effective treatments there with this. And yeah, so like evoked pain, hypersensitivity can go down. That's only a small part of what their overall problem is in life, right? And so we're looking for patterns in the brain that are related to chronic pain and essentially maybe directly related to the sort of fear and avoidance, right? And then those would be new targets. And yeah, our back pain study is an intervention study in 150 people, where we're trying to do that, and we'll see how it goes. I mean, we got the study done, and we're working on the brain data. But yeah, so like in some cases, this signature can be useful, right? If it's like evoked or immediate pain, right? But if it's sort of ongoing pain and fatigue, we might need new targets.
Have very strong placebo, but have you looked at you talked about the whole behaviors? Have you seen if that has an effect on the
So yeah, those so we can I guess the thing is we can manipulate people's beliefs in many ways, and maybe what they consciously support as a belief may or may not be a critical factor, right? But, you know, yeah, so like all these placebo studies are attempts to manipulate beliefs in that way. Right? And they do, you know, people do expect less pain. But I think, you know, I think the sort of standard sort of amount of reinforcement and way of doing it isn't, you know, it's not really enough to have a big effect. If you take like a fake cream, just to give you a sense, like people react to it really differently. One of my first participants said, "Oh my gosh, creams, I get rashes. I can't deal with rashes. It's gonna, you know, without basically, right? You know, and so there's a lot of variability in how people respond. They use these creams all day long, and so you're not capitalizing on sort of very consistent, you know, changes in expectations because people vary, and you're not necessarily capitalizing on a sensory experience in classical conditioning that's uniquely associated with inland. So, and I might be being unclear there, but there's like, you know, there's different ways to tap into something that's kind of powerful. That's why I think this symbolic conditioning thing is interesting, because it's avoiding some of these sort of low-level learning effects, which might actually go in the opposite direction as placebo effects. Like you prepare for pain, you release opioids, you experience less pain. So there's that, right? And instead focus on this, you know, sort of conceptual mindset-based manipulation that seems to have much larger effects. So what am I saying? Devil's in the details, but maybe, right? In terms of which things actually work, right? And we have to maybe work backwards to figure out, you know, why exactly.
That makes sense.
Yeah, so here I kind of went through it fast, but it's basically symmetric. Yeah. So if you're expecting, you get a high pain cue and it's worse than expected, you update more. Or low pain cue and it's less than expected. That's right. Our claim here is that you update less, right? Yeah. And that kind of effect is not, to my knowledge, in any of the reinforcement learning models I've learned about. There's many variants of models, but like this kind of confirmation with initial expectations. You know, it's different. More generally, I'm really interested in our capacity to discount our experiences, right? Or to interpret the same kind of evidence in very different ways, depending on, you know, what's expedient for me or how it's different. That's the direction we want to do more.
Just out of curiosity, so it might not be in reinforcement models, you know if it's in reinforcement. So for example, if you're rewarded with food and it's like, you know, high, like M&Ms, oh great, you get more M&Ms, is that, you know, as opposed to less to do? No, is there a comparable thing, or do you know whether the people look at it?
You know what? I don't know of anybody else who has fit that model either. You know, there's a million papers. I don't know them all, right? But yeah, right. But it's interesting. I just like what I've seen is you know, differential learning rates and things like that for rewards and punishments, which is pretty typical, right? You know, and state action value. There's other like, you know, P value and action value. But not a sort of a confirmation bias parameter as far as I know. So it would be really interesting to actually fit this kind of model with other kinds of data.
Yeah, so
right?
it's pain special.
Yeah. And one thought too, I'm sure there's a lot of things that like are that it would work and it wouldn't work essentially, or this effect is operable. But I think one of them is that if it's really clear, if it were like, you know, you get an M&M or you don't, right? There's not very much ambiguity, you know, and so your sensory evidence is very strong, right? You're required, like, you know, the posterior is going to be focused on the evidence. Here, I'm constantly surprised by, I think, how pain is kind of tangible. Like, it really is, like, you know, I think it's weird because, you know, clearly people are really reliable. It's more intense and it hurts more and they're really good mostly. But also, you just kind of think, like, what did I feel? I don't know. How bad was it? Right? So I think that might be part of it too, right? So it wouldn't necessarily work in every learning paradigm, but where you either get a cookie or you don't.
Great. Thank you very much.
Thank you.
For the wonderful
Yeah.